president of the Sister Science Club, and I'd like to thank a few people here for being here. First, Angeline and Belfry staff, really appreciate it. We have a few board members besides myself. Don Udinger is up in the back filming this thing. Dr. Larry Price is out here somewhere over there. I don't know if David Ellis is here or not. There he is, snuck in the back. And Joe Sherry. Our secretary, Gail Todd, is around here, over there in the corner. And Joyce Bevlins is somewhere. So she's our STEM teacher. Oh, right there. Okay. I can't see here. All right. We are looking for some new board members, and Gail has some applications if you're interested in doing that. And then I should mention Peyton Mayer is our sound person. And there's one more announcement, which is that there's a STEM night at the Sisters Elementary School on February 26th from 5 to 7:30. And you're welcome to come and see the various things that are going on in the schools with STEM and STEM-related activities. So, within our lifetimes, biology has crossed a remarkable threshold. We moved from discovering that DNA encodes life instructions to sequencing it, and now to deliberately modifying it. This transition from observation to intervention is the subject of tonight's program, genetic engineering and gene therapy. Humans have been modifying genes through breeding, but direct DNA-based methods began in the last quarter century, in the quarter century prior to the 2000s. Before the century, the end of the last century, when molecular biology, it was still largely descriptive. Genetic biologists identified mutations, mapped genes, and correlated genotype with phenotype. Today, the field has evolved into something far more operational. Genomics guides diagnoses, stratifies patients, and increasingly shapes therapeutic strategy. Dr. Francis Collins, former NIH director and co-leader of the Human Genome Project, once referred to DNA as the language of God. Whatever metaphor one prefers, the critical point is this: we have learned to read the language at scale and began editing it. This past weekend, I accompanied Sisters High School students to the Fred Hutchinson Cancer Center. Over 2.5 days, they learned basic skills for DNA research, micropipetting. If you think of a turkey baster, you suck in the juice and you squeeze it out. That's what pipetting is, and they make these devices, though, where you can bring in microliters at a time and deposit them at a time. It's very interesting. And gel electrophoresis, that's making a gel with wells in it where you put DNA samples and then apply an electric field across the gel to make the DNA move according to their size. It's a very, very interesting thing. They also did some bacterial transformation work, examined blood cells. They toured the labs where they were introduced to advanced imaging tools such as electron microscopy and flow cytometry. And they talked to the professors and PhD candidates about their career paths. For more than 20 years, this annual visit has been supported by many of you in the audience, as well as the science club, and I really want to thank you for that. What stands out is how normalized some of these techniques have become. The tools that once defined graduate level research are now part of advanced secondary education. Beyond oncology, DNA-based technologies now influence multiple domains, including forensic science and to reproductive medicine. Ancient DNA analysis is one of my favorites. Ancient bones have been dug up, DNA extracted and analyzed. And what this has led to is a refinement, in some cases, correction of our understanding of human migration, the spread of European languages and evolutionary history from earlier hypotheses based on archaeology and linguistics. Much of this modern capability traces back to the Human Genome Project, when we first drafted human gene sequences that was announced in 2000. We moved from studying isolated genes to working from a comprehensive genomic reference. That milestone paved the way for precision medicine and molecular diagnostics. Now, in the 21st century, advances in gene editing, particularly tools such as CRISPR, have made targeted DNA modification far more feasible and precise than anyone thought imagined. Without preempting Dr. Scher's discussion, what is groundbreaking is that scientists can now alter specific genetic sequences in living cells with therapeutic intent. That capability underlies emerging treatments for inherited disorders, engineered immune cells for cancer therapy, and regenerative approaches to damaged tissues. It also raises serious questions about safety, regulation, long-term effects, and equitable access. Far from being abstract debates, they are active issues in medicine and public policy. To guide us through this landscape, we are fortunate to have Dr. Scher, whose career spans more than 30 years in gene therapy, regenerative medicine, oncology, and diagnostics. Educated at the University of California, Los Angeles in the Scripps Research Institute, he has worked at the forefront of biotechnology from its early growth through its current translational maturity. Dr. Scher advanced early oncolytic virus technology, a powerful treatment for cancer, at Caledon. At Asthmatics, he helped bring an asthma treatment device from pilot studies through regulatory approval. As a consultant, he supported the launch of generic cancer diagnostics at Genomic Health and Janssen Diagnostics, translating molecular insight into clinical tools. Today, as founder and CEO of Haptics, he leads the development of innovative cell therapies for liver diseases. He advises the Stanford SPARC program and directs the StartX MedLab Cooperative. He is the author or inventor of more than 30 peer-reviewed publications and patents. His career reflects the full arc of modern biotechnology, from experimental platforms to clinically developed therapies. He's also a pilot, a native of LA. He's been coming to Central Oregon since the 1990s to visit family, and now lives outside of Sisters with his wife Melinda and dog Wally. We are living in a moment when biology is becoming an engineering discipline. The ability to intervene at the genetic level carries extraordinary promise and serious responsibility. Tonight's lecture will explore both. Please join me in welcoming Dr. Scher.
And it work? Is it working? Yeah.
Yeah.
Okay. All right. Just want to make sure I get it secured so that it doesn't fall off in mid-sentence or something like that. New fancy technology. Thanks David for that wonderful introduction. And I think you've covered it. So is there any questions? I'd be happy to answer them. Seriously, no, that was wonderful. You gave a really great kind of broad view of the kind of stuff that I'm going to cover in a little different angle on it tonight, and in a little more depth in some of those things. Let me see if I got this on. I guess we're Is the audio okay in the back?
Yeah.
And these Sisters Science Club talks are awesome. I was here last month to hear about dark skies and their importance in our lives from Rima and Scott, which was an awesome talk. And I'm honored to be able to be here and talk about something that's near and dear to my heart, which is gene therapy, which we'll get into a little bit. But before we do that, I want to shout out to the Sisters Science Club for their support of experiential learning at Sisters High School for this kind of stuff, because I've seen what it can do. I've seen the effects of it, and it's really impressive. These two young ladies here are a great example. Jessica there on your right came to Hepatics to do an internship with me one summer several years ago. She was a freshman at the University of Washington, didn't know what she wanted to major in, really didn't know where she wanted to go with her career. And so we spent the summer doing some pretty sophisticated experiments. We got into flow cytometry and things like that, and it clicked with her. And she knew by the end of the summer what she wanted to do. She went back to the University of Washington, majored in biology, spent 3 years working at Fred Hutchinson as a research technician, and now is in the middle of her PhD studying immunology at the University of Chicago. Her sister on the left up there, Sarah, was a slightly different case. She came to do an internship at Hepatics as a junior in high school. Very smart, very intense, focused young lady. And so she came in kind of knowing this is what I want to do. And the effect of having this experience in the lab was to confirm that for her. And so she went on, I think she majored in biology at Tufts, and she's now in the middle of getting her DVM degree at UC Davis. So, I've seen firsthand what this can do, and I was really impressed that the Sisters Science Club could come together to support Sisters High School in this, and I hope you all will help with that effort as well, because I think it's very, very worthy. But a little bit more about me. David introduced me, but I'll say a little more. Like he said, I've been in the biotech industry for 30 years after my postdoc. In the early parts of my career, I spent a lot of time engineering genes and viruses. So we'll talk the kinds of things we're going to talk about tonight. But about 10 years ago, I founded Hepatics to develop cell therapies for liver disease, which is an enormous problem worldwide for which there are not very many good solutions. And cell therapies are kind of gene therapy adjacent, and you'll see a little bit of why that is as I go through the evening. But I've found in the biotech industry really a passion for trying to take these great scientific insights that fill journals and libraries and try and figure out how to move them forward to products that actually can help people. So that's a lot of what we do. My core team is up there. They're awesome, smart people. The folks clustered there on the right were supported by a wide array of really very talented, smart people, from hepatologists at the Mayo Clinic and Mount Sinai School of Medicine, UCSF, to stem cell biologists and business people who have run cell therapy companies. So it really does take a village to do this. It's a lot of work goes into bringing these drugs to people. Today, I'm going to talk a little bit about history of genetic engineering and cell and gene therapy, which is often abbreviated CGT. Then a little bit talk about some examples of gene therapy and how they've evolved going forward to try and illustrate how these things are developed, and then peek out into the future and see what this might look like going forward. And there are some themes that I hope will come through in this. One of them, that we humans are inveterate tinkerers, and we've actually been doing gene engineering for a long time, and that this is a real process with warts and failures and iterations. You might be surprised to know it, maybe not now that David said it already, but we've been genetic engineering for centuries, actually. We just used to call it selective breeding. And Yeah, that's what we used to, I mean, but it accomplishes the same thing as genetic engineering. And so I've got an example here from the dairy industry, which we know a little something about in my family. The fellow there in the bottom center is E.A. Stewart, who founded Carnation Farms up in Washington, the home of contented cows, if you remember that. And he realized in the early 20th century that genetics could improve his herds, and he could control that and thereby improve his yields, milk yields, which obviously would be a good thing for his business. And in 1918, he bought this calf that's shown in the top right there. Carnation King Silva is the name of this calf. He paid $160,000 for this in 1918, $2 million in today's terms. Just an outrageous sum of money for a calf. But he knew that the calf's father had sired high-producing cows, and the calf's mother was a high producer herself. And so this calf could transmit those traits to its offspring. And in fact, that's what happened. As you can see in the bottom left there, that table, following his selective breeding protocols, they increased their milk yield 30%. So it does work. But let's step back to the 19th century to talk about how the foundations for this actually happened. Who here took genetics? Okay, there's enough people, so I'll have to be honest about this. Anyway, this is like, you know, conceptually something that is difficult for some people to grasp, including me. Gregor Mendel was a priest in Austria in the 19th century, and also a biologist, and very curious about heredity. And back at that point in time, biologists thought that heredity was kind of one of these fluid things. You got a little from here and a little from there. It's kind of like spirits that could mix and match. And so he bred pea plants because they had, you know, very definable properties that he could trace. And his theory of heredity was, you know, kind of game-changing, provided a foundation on which the rest of molecular biology of the gene has been built. And that had two aspects to it. One is that heredity wasn't a fluid, little this, little that, immeasurable thing. It was discrete, it came in discrete units. And he also defined the math by which that could happen. And so upon that foundation, the rest of the story is built. That all culminated across the succeeding decades with the publication by James Watson and Francis Crick of the structure of DNA shown over there on the right. It's a double helix with a very repeated regular structure. They published that, and they also published a model for how that structure could produce two important things. One, two important things that would sustain heredity. So quick molecular biology lesson. Actually, David had a great slide earlier about, it was a better helix than my helix he's here. So DNA helix up there on the top, the repeating units are represented by letters here. And so what the two things that were important about that was it gave the cell a way to make an exact copy of the DNA for each of the daughter cells. So when a cell divides, that parent cell could make a copy of DNA for each of those daughter cells. It was an exact copy. So you can pass on traits that way. And the second is that it could be read like a book. And you can see that in the middle there. And what that did was the cell's machinery could read this book of life, as we could call it, and make proteins from that. And proteins are the structure of our cells. They're what composes our bodies mostly. They give us our hair color, they make energy for our cells, they digest our food. So the ability to manipulate this structure at will is incredibly powerful because it allows us to then manipulate the proteins, which then allows us to manipulate the cells. So it's just an amazing advance. There's your molecular biology lesson. And what emerged out in the 1970s and 1980s in terms of molecular genetics was truly it was clear it had the power to be transformative. But like any new technology, there was hype associated with it. So both hope and hype up there in the top. Transformative cures for medicine. One-shot cures and so forth. Lots of hype, but we humans, being humans, often would take that and twist it in new directions that are negative. Our brains actually react to negative information more strongly than positive. That's just the way we're built. And so that's why newspapers, run negative headlines. And we're seeing a lot of this actually with AI these days. If you kind of think about the parallels, and they're saying, on one hand, it's going to allow us all not to work anymore. And on the other hand, it's going to make humans extinct. So, same thing happened with genetic engineering early on. The powers that be, the stakeholders, the main people who were driving this, like Paul Berg at Stanford, realized they could see it if they didn't do something in terms of reassuring the public that this could be used safely. There would then soon be political reasons why it would just be either banned or go out of control. So they were smart enough in 1975 to call a short halt to all of the recombinant DNA experiments, gather at the Asilomar Conference Center in Pacific Grove, and have a conference to develop a set of rules in order to guide recombinant DNA experiments and the technology development. They created a committee to approve all of these experiments called the Recombinant DNA Advisory Committee. And the system actually worked pretty well. It gave people a way to develop this without wasting time on one hand and also doing experiments which could create a negative public image for this. And we've benefited by it. And so actually it was in place until just about 15 years ago. Out of this have emerged, literally hundreds of products, enzymes to make our whites whiter, crops that are disease resistant and more nutritious, and medicines that couldn't have happened another way, like human insulin, which wasn't available to people with diabetes before this. And companies like Genentech, Amgen, and Genencor were founded to do this, and they now employ literally tens of thousands of people worldwide. So it's been quite a run. Looking more specifically at cell and gene therapy here, just over the last 20 years or so, there's been dozens of FDA-approved therapies in this class. Just in the last 3 years, 13 new gene therapies to treat a whole variety of diseases, including Duchenne muscular dystrophy and sickle cell anemia. These are FDA-approved ones, and then there's thousands more behind this in the cell and gene therapy class that are aimed at treating an amazing array of human diseases in ways that weren't possible before. So it's really still a flourishing technology, very, just with a lot of promise that's still yet to be realized. So let's shift gears a little bit. I'm going to start talking about different flavors of cell and gene therapy. One, gene replacement, and then engineered cells, and then the ability to actually do that gene engineering right in our bodies. And it'll illustrate some interesting things about how these things can evolve. So gene replacement was the earliest version of gene therapy. And so think about in our cells, one of the cases we'll talk about is a young man who had an ornithine transcarbamylase deficiency. So this is an enzyme in his liver that's not functioning properly, he's got a mutation, and he's sick. And so how could you treat this? There wasn't another way to treat it. There wasn't a way to get that enzyme in his liver. So gene therapy offers a solution. What if we could put back into his liver cells a functional copy of that gene? It should work and it should cure that disease, right? So that's gene replacement. Then the next step you could take beyond that is to actually take cells out of the body, engineer the DNA in those cells, and then put them back. And that's going to be CAR T therapy, which we'll talk about. And then finally, we'll talk about gene editing, where we can actually do this inside the cells in the body without taking something out and putting it back. We just actually just do it right where it is. So, if you'll bear with me, I'm going to explain gene replacement in some detail, and this will hopefully support some of the other things we're going to talk about. But the basic idea here is we isolate DNA. This picture down here in the lower left is isolated DNA, probably in alcohol. We can take that and snippets of it, of these, gene X, whatever it is that you need, put it in a DNA backbone, which is that black circle up there with the blue arrow. And what that backbone does is it actually controls how that gene is expressed. So we're putting onto that gene its control circuitry. And then next is to package it into a vector, which is going to be the thing that carries that back to the cell and places it in the cell where we want it to be. And there's usually two varieties of these vectors, viruses and chemical coding, basically, on the DNA. Viruses are a natural choice for this, because that's what they do. If we get the flu virus gets into our lungs, it attaches to a lung cell, and it injects its DNA, or RNA actually, into the cell, and then the cell processes that and makes new viruses. So their job is to carry DNA or RNA and put it into cells. So that's what we've done, is engineered these viruses to carry our engineered DNA into the cell, where the cell's machinery translates it into a protein, hopefully the repaired protein that we want to replace, which is now in the cell and replacing that lost function. So hopefully that all makes sense for you. What could go wrong? I've done this. It's like, what could go wrong with this? It's obviously going to work. Unfortunately, there were problems. So I'll talk about these two cases to just illustrate. DNA is pretty inert. These viruses that we're using were thought to be relatively innocuous. Jesse Gelsinger, shown here on the left, was a teenager who had that ornithine transcarbamylase deficiency. And so, like I was just describing, the idea was, well, we'll just engineer one of those, the functional version into an adenovirus. It'll go to his liver, he'll have the functional enzyme, we're good. Adenovirus is, we all have adenovirus antibodies. We've all been infected by adenovirus. It was thought to be pretty safe. So they did this, and unfortunately his liver failed and he died in 4 days. So it was a real shock to the gene therapy system, and in particular for me, at this point in time, I was working for another company actually engineering adenoviruses to treat prostate cancer. And so we were very closely watching this, and it was not good news when it happened. The other case here is bubble boy disease. You can see the kids have to exist in a spacesuit basically because they have no immune system. And it's a deficiency of a gene in their T cells that allows the cells to attack germs. This was done in France, and these scientists engineered that IL-2RG gene into another kind of virus called a retrovirus, which is related to the HIV virus. And this virus has the property of actually inserting itself into the DNA of the cell. So it gets into the cell. Adenovirus just kind of floats around and expresses its proteins. Retrovirus actually becomes part of the cell's DNA, which is good in the sense of that it will always be there, it will be passed on to all the subsequent cells, should be a permanent fix. They did this, they tested it in 20 children, and it worked. I mean, these kids their immune systems were reasonably restored. They could be out of their bubble. But the downside was that 5 of them got leukemia from this treatment. And that was because that retrovirus had integrated itself in the DNA in a cancer-causing gene. And so it actually turned on a cancer-causing gene. Another bad outcome. That was one of the feared outcomes in gene therapy. So between the two of them, these two episodes basically sent gene therapy into a tailspin for about 10 years. Fortunately, scientists continued to work on it anyway. So there's, inveterate tinkerers, and there were people who still wanted to do gene therapy to work. And this is one of those cross-fertilization sort of moments in science. They were working on adenovirus trying to fix it. They were looking at retroviruses trying to fix them. But somewhere over, the adenovirus scientists who were studying just adenovirus because they liked adenovirus had found that there was this other little virus that kind of was a passenger in those infections called adeno-associated virus. And it had some useful properties in the sense of being a vector for gene therapy. One of them, it was basically pretty benign. It didn't integrate in the DNA. It didn't cause an immune response. And its main drawback was it's pretty small. But it had a lot of properties that were very useful in terms of being a vector for gene therapy. Our next example, excuse me, is looking at Duchenne muscular dystrophy and how this all came together to treat that disease. I don't know, could I get a bottle of water, David? Pardon me. I'm getting over a cold from a couple of weeks ago and my throat's still a little bit scratchy. Another viral disease. So Duchenne muscular dystrophy is an X-linked recessive gene, which means that only mothers can pass it to their sons, and it's a broken dystrophin gene, so a mutated dystrophin gene, which is in the muscle. And so what happens is these sons who inherit this actually get this muscle wasting disease and die early. And you can see that in that picture over there. Thank you very much. But it's another, ideal test case for gene therapy. It's replacing a single gene that's broken. The results, if you can do it, are dramatic and they're permanent. And so persistence pays off. The main trick here, well, there were actually 3 tricks, but the one illustrated in the top, that top circle, is that the scientists had to figure out how to engineer down the size of that dystrophin gene, which was pretty big, to fit it inside that adenovirus, and they managed to do that. Then they figured out how to make a lot of that adenovirus, and then they figured out how to treat people with it effectively. And the result was that Elidivus was approved 2 years ago to treat this disease. So you can now treat Duchenne muscular dystrophy with a gene therapy product. So it made it across the finish line, which is an awesome achievement. And a call out to Oregon, Sarepta actually started as Antiviral Inc. in Corvallis 30 years ago. So it's pivoted a little bit, changed its name, but kudos to Oregon for producing a company that could bring this across the finish line. So what did we learn from this? We learned that you can replace genes and treat previously untreatable diseases, that there can be side effects from these therapies. It was a question we didn't know the answer to. And the good news was we could fix those problems. So this took lots of iterations, it took lots of time, it took failures that people were willing to overcome. So science is a process. It takes time and it takes a process. Next example, we'll look at engineered cells. So this is CAR T therapy, and it's kind of a convergence of 2 different fields in order to go to kind of the next level of gene therapy, so to speak. And I'm going to set this up with a discussion of another way that various science disciplines feed one another. So before we could do CAR T therapies, a couple things had to happen. One, back in the first part of the 20th century and before that, scientists discovered that our bodies actually can mount an immune response to cancer. Great news. So that discovery was part of the equation. The next part was bone marrow transplantation, which was started being implemented in the 1950s. And in that particular scenario, the cancerous bone marrow is removed from the patient and it's replaced with normal bone marrow. And the cells engraft there. And so the important advance out of that is cell engraftment or cell transplantation. I have a mouse here. This is one of the crossovers to cell therapy as well from hepatics. That mouse actually has a humanized liver. And the panels below that show with different stains those human hepatocytes engrafted in that mouse's liver. This is something we work on at Hepatics. We're working to engineer cells to be able to replace engineered cells back in the body. So another process slide, another explainer slide here. This one about CAR T therapy. So if we look starting down here, actually in the lower left, as I mentioned in the previous slide, our white blood cells are able to recognize and attack cancer cells. That's great news. We look at the next level up here. Unfortunately, the cancer cells have learned how to turn off those white blood cells. Bad news. So a whole other branch of science, immuno-oncology, sorted out how that happens. How do these cancer cells turn off the immune system? And they've created a whole set of drugs that actually do that. But that's also translated into the CAR T therapy. So that middle panel, that circle with the slash in it is over that molecule that cancer cells use to turn off our white blood cells. So the trick in CAR T therapy, and what the CAR is for, is chimeric antigen receptor that's found in T cells, is to engineer that receptor so it can't be turned off by cancer cells. And that's what they did. So the circle over there, the process is to isolate white blood cells from the patient, engineer that antigen receptor so it can no longer be turned off, grow up a whole bunch of them, reinfuse them into the patient, where they'll engraft and attack the patient's cancer. So this all should work on paper. And happily, it also works in people. So Emily Whitehead over there on the far right was the first person to be treated with CAR T therapy. She had the diagnosis of acute lymphoblastic leukemia at age 5, and that's basically a death sentence. Those kids don't survive. She received CAR T therapy. She's now 13 years cancer-free and a healthy college student. And just over in the intervening time, there are now 7 FDA-approved versions of CAR T therapy attacking 6 different types of cancer. And where most of these patients, it was basically, they were going to die. For most of them, it was a death sentence. Now the survival rate is around 2/3. So just an incredible advance, a big impact in oncology. And a further illustration of how these things evolve, they're now looking at how to apply the same technology to treat autoimmunity and fibrosis, which are both additional huge medical needs out there. So another technology that's evolving forward. So where do we go with it next? What else could you do with this? As I mentioned earlier on, with gene replacement, we take DNA out, manipulate it, put it back. With CAR T therapies, you take cells out, engineer them, put them back. So could we just skip the take it out and put it back part? And that's what this next evolution is. If you could do that, you could basically fix any genetic disease right where it is in the body. You could even tailor the human genome blueprint to have it be what you want it to be. And then the question is, would that be a good thing to do? CRISPR is the technology which has been invented to do this. So CRISPR stands for clustered regularly interspaced short palindromic repeats. I'll test you on this later, so remember that. But in a further example of cross-fertilization between fields, this arose from observations back in the 80s by bacterial scientists who bacteria have their own viruses they have to deal with, and they noticed that some of them could survive this, and these repeats were involved in that. And it turns out that the CRISPR-Cas system is actually kind of the bacterial immune system. And so some smart people worked this out, that they could isolate that system and engineer it in certain ways. Dudna and Charpentier won the Nobel Prize for this in 2020, and used that same system to edit basically any place in the genome, and make any kind of change you'd want to make in those cells. And a test case for that is sickle cell anemia, which is another single gene disease, pretty widespread. But it's a case where there's one amino acid substitution in hemoglobin, which is what makes our red blood cells red, carries the oxygen to our bodies. And in that non-functional hemoglobin, because of that, these patients get these pain crises, they get organ damage, they get infections. It's not a disease you want. But using this technology, which I won't go into the details of, but it's basically a small protein and a guide RNA is what they call it, that can be introduced into the cells right in this person's body, Marie there in the bottom, and it finds its way to the right cells in her bone marrow, makes the edit in those cells to correct that, and then she now has normal hemoglobin that can alleviate her disease state. And Casgevy was approved 3 years ago to treat this disease. So another successful example of bringing one of these novel therapies across the finish line, and how fast they were able to do that is the other important lesson here. So CRISPR provides some interesting lessons for us, both in that we can do this in such a flexible way, but it also can be done quickly. And a case to illustrate that is here, and this was just last year, baby KJ in Philadelphia. And in this case, this baby was born with another single gene defect called CPS1 deficiency. And that's an enzyme in its liver that is misfunctioning, and basically the only treatment for this infant would have been a liver transplant. And the other bit of information relevant is it's a very rare disease. There's only about 3 of these children a year born in the US. So this was, it's a desperate situation for these parents. There's no approved therapy for this. There's only 3 kids a year, so nobody's going to make a therapy for this. But scientists at Children's Hospital of Philadelphia had the idea that they could make a custom CRISPR therapy for this baby and treat that baby. And so they did that over a period of a few months, treated the baby. It actually worked. So here you have baby KJ's age 6 or 7 months is already treated with this kind of a technology. It was just an incredibly fast development process. So you might say, well, Eric, that's just not relevant to everything else. There's only 3 of these a year. How can that be useful for us to know? Well, in fact, these rare genetic diseases are not so rare. There's 7,000 plus of them in the U.S. That's the diseases. That's not the people. Most of these are single gene defects, so they're potentially fixable with this kind of technology, and almost none of them have an approved treatment. But collectively they have a large economic burden, and if you have a technology which is easy to customize, can be done quickly and inexpensively, then maybe you can treat these patients. And so what it gives you is like turning medicine kind of on its side, to look at it a different way. Instead of making therapies that you treat thousands of people with, you're actually making a therapy you're going to treat a few people with, but you can do it quickly and cheaply. So it's just a new way to look at medicine and an example of how technological advances can change the way we do things. So where could you go with this? You know, where else could you go with this? If you can, you know, so baby KJ was a baby, you know, 8, 9, 10 pounds, lots of cells. So we fixed a few cells in this baby's liver, and it's great, you know, it helped with that baby's disease. But baby KJ could probably, you know, when he grows up, pass that same CPS deficiency to his children. But what if you could treat embryos where there's only a few cells or 3 cells and fix all of the cells? Then that baby, when it's born, has that fixed in all of its cells, and it forever is not going to suffer from that disease, nor will any of its children. That's sort of a, you know, a dreamy thing out there, but you know, then you're kind of looking at, are you going to have designer babies that you can make there? There are good reasons to do this. It would be eliminating these kind of diseases like CPS1. There are some maybe not so good reasons to do this that are over there. You know, designer babies are maybe a little controversial. And that article that I've put the reference to down there in the lower right is actually a very interesting article that talks about this kind of technology and the situations that are involved just from a few months ago. So you're saying to yourself, but Eric, this is just theoretical. It's not. He Jiankui was a very ambitious young physician scientist in China. And unbeknownst to the world, and apparently the Chinese authorities as well, he was editing embryos. And the motivation here was he was editing in these embryos a gene called CCR5, and that gene is responsible for the entry of HIV virus into the cells. And this embryo's mother was HIV positive, so the thought is, I can immunize these babies against ever having HIV infection, which is a positive outcome if you can do that. He was successful with 3 embryos. One of them had the complete knockout he was desiring. All 3 of them were born as healthy babies, and they're still alive, so they're healthy as far as we know. And for all of his efforts, he spent 3 years in jail. And kind of the pluses and minuses there, the pros was it would do that, it would block HIV infection, but the minuses are we really don't completely know what CCR5 gene does. So what's going to happen to these kids when they grow up? Are they going to have a health problem because they're lacking that gene? It's not there for no reason. Others are, the CRISPR technology actually can make changes in the genome besides the targeted change you're trying to make. So did he make changes in those kids' genes that he wasn't expecting to make? So, there's questions like that that needed to be answered. And so when this all came up, what do? Do you go back to the drawing board and say, we're going to eliminate this genome, germline genome editing, as it's called, or are we just going to ban it? And fortunately, that's not what happened. So this kind of harkens back to what happened at the Asilomar conference in 1975. The stakeholders in this particular branch of science said, we're going to stop. We're not going to do genome editing for a little bit. And we're going to get together and put guardrails around this, decide on what rules make sense in order to responsibly advance this technology. And then, so some of the primary national and international bodies who chime in on these things, like WHO and the National Academy of Sciences, wrote reports on it and concluded that no, we don't want to ban it. We need to have a way of moving this forward responsibly, and some of the criteria for doing that are over there on the right. And you know, in my opinion, I think, and a lot of people would probably agree, is you ban germline genome editing, and that's not going to keep people from doing it. Somebody's going to do it anyway. So you may as well establish a system for moving this technology forward responsibly. So another Oregon call out here at OHSU, Dr. Paula Amato, who's a OBGYN there, is one of the leaders of this germline genome editing field. And I had a chance to talk with her and pick her brain on this whole subject. Very interesting. And so there are good reasons to do it, but proceed with caution. So that kind of brings us to the bleeding edge of medicine and genetic medicine. What I hope that we'll take home from this is that we humans tinker with stuff. We've been doing it for a long time with genes. We find new ways to do it, and we're going to continue to do it. So I think we may as well do it the right way and embrace that. The other is that all of these took a long time to accomplish. None of them appears magically out of some lab in the span of a few months. I don't think any of them happened in less than 20 years. So science and technology development are a process. They're not a thing. They happen because people are engaged in it. It's a verb, it's not a noun. And then finally, if we follow that process, if we do these kinds of things I've been talking about for the last few minutes, we can come up with real-world solutions to real problems. And I think that's an awesome thing and a great reason to be involved in this, as I am. So, I'll thank you for giving me your ears for a little while. I hope you found that interesting. I love this stuff, and I'd be happy to dig into any of it with any of you. My contact info is up there, and thanks for listening. Yeah, actually I am. I was going to take this off, but I guess I'm going to keep wearing it. Questions?
Yeah, I'm curious what we wait for the mic.
Hi, great talk. I'm curious if you could share with us your thoughts regarding the intersection of computation, AI, and modeling, and how that's changing this field. In other words, can more be done computationally before some of these experiments on humans that in some cases haven't gone well? Is that a fertile ground for discovery?
Yeah, that's a great question. So how can we use computational and AI methods to try and sort of predict what's going to happen? And basically it just shortens the experimental cycle. It doesn't eliminate anything, I don't think. I think it can do that. We're working on it, and other people are working on it. Right now, I think at least the AI type of approaches are sort of situationally useful in places like manufacturing and clinical trials. To get to, I'm going to make this change in this gene and it's going to happen, and it's going to have this effect, is still out in the future, because there's too much we don't know about biology. Kind of akin to the CCR5 example where you can knock it out, it should be okay, but we really don't know what that gene does, and until that's defined, it's hard to make predictions about what's going to happen.
Dr. Scher, thank you so much for being here and for your talk. So, a couple of the examples you gave were single gene maladies, sickle cell anemia. And I was just reading about AI or large language modeling research that we're identifying, I think, and this is beyond really my expertise, but I think what they were saying was that frequently proteins are produced by base pairs that might be thousands of pairs away from each other. And that, so not a single gene malady, but multiple and not right next to each other, right? The way I would think, I would organize things. So I wasn't involved in that. But my question is this a possibility that artificial intelligence might bring us to solutions to multiple gene maladies and using CRISPR and other technologies to solve those problems?
Yeah, that's a great question. I think theoretically, yes. Again, it requires the information for those models to use to make those predictions. And until we, and that'll improve over time. So I think this will begin to happen. But right now, doing more than one gene, it's that kind of exponential effect that you get 2 and it's 4 times as hard, and you get 3 and it's going to be 9 times as hard, that kind of thing. So I just think the systems aren't, the data, not enough data is there to make use of those kinds of models to predict accurately what's going to happen. But I think it can happen someday.
Thank you.
Yeah.
Thank you. So I want to follow up on Davis Ellis's, comments, which are very pertinent. I'm another Dave. Like, this is another Dave over here. My question is that I've spent a career in biomedical research, including NIH. Francis Collins is the director, and I'm a veterinarian. And I have witnessed tremendous advances in biomedical research and application to human medicine. And I really liked your examples that you showed here. And my question, which I think is very pertinent in today's society, is that I still believe that selected animal models are critical for the advancement of human medicine. And that's been my focus. I focus on human medicine, not animal medicine. And with your expertise, do you see animals continuing as a vital importance to the non-animal technologies? And I'm saying that, and I'm going to give you a political thing. This administration wants to wipe out all animal research in less than 10 years. I appreciate your comment.
Sure, yeah. I've done lots of animal experiments. We do them at hepatics. Yes, they do want to, it's been across my entire career, it's been this reduce and replace kind of thing with animals and trying to, and I think that's reasonable. It's like, don't use more than you have to. They are expensive experiments to do. But they do provide a level of information that you can't get from an in vitro model, even the organoids that are out there. And we're doing some organoid type experiments. How do you model cells and grafting in a tissue like that mouse with the liver there? In order to function in that 3D environment, we don't have the ability to do that without animals. Plus, there are toxicology effects that happen on whole animal physiologic systems that you can't model other ways, and there's not going to be a computational program that's going to do that anytime soon. Because again, we don't have any way of predicting what that's going to be. So I mean, I think we're basically on the same page. I agree in terms of trying to reduce as much as you can those animal usage. But, you know, if you go over a certain level of reduction, the risk goes up with the humans that you later test those things on.
So, thank you for your presentation. I'm a lawyer, I'm not sure I understood anything you talked about. Except that it's apparent that this is a powerful tool with tremendous potential for positive effect on people who need what you can address. Is there anything about this powerful tool that keeps you awake at night, that causes you to worry, that we also should be considering as we look at setting up the guardrails for the use of this tool?
Me, no, not really, because I mean, I love biology. I still am just blown away by biological systems, and I wanted to work that into this, but it would have taken too long. I don't know that we have the ability to predict what's going to happen by making, you know, how would you make a kid smarter? I don't know. And I don't think anybody knows, and because it involves too many factors, and we just don't have the information to make that happen. People are going to try to do that, and you know they will end up, you know, maybe moving the needle on some measure of that, and then they'll find out there are a lot of downsides to it that they didn't want, that came along for the ride. So I just don't think, you know, there's going to be a lot of failures before anybody gets to the point where, you know, you could, I guess the fear thing is the superhuman, engineered superhuman. I just don't see that's ever going to happen in anybody's lifetime in here.
Other questions?
Hi Eric, great talk, thank you. I have a question about, you work with individual variations in genes. What about, is there any work on epigenetic modification of genes with epigenetics, basically?
Yeah.
That's a huge factor in turning genes on.
Yeah, and it's sort of the multiplier that I think makes the engineered human almost impossible to even conceive of right now. And that's an entirely different 1 hour lecture. There's a whole series of companies and labs working on that. I think it's fascinating stuff, and I try to follow it as best I can, but it would have been too much to add tonight. Yeah, totally.
Hi, great presentation. It seems like there's at least much more awareness, maybe incidence, of autoimmune disorders. And I saw autoimmunity as one of the areas of expanded research. I wonder if you could speak specifically to that.
Yeah, so I was reading an article this morning, and there is a company out there who is taking CAR T therapy to, so the essence of the problem is to target that CAR T cell, which is basically sort of a, you know, a guided missile kind of thing, if you want to think about it that way, and target it at that cell that's causing the autoimmunity. It's oftentimes T cells that do that. So if you can figure out which T cell is going to do that, there's some, you know, immunologists have ways of figuring that out, then you can engineer that CAR T cell to attack the immune cells that are causing the autoimmunity. Does that make sense? Yeah. I think this gentleman over here, he's been raising his hand for a while.
Thanks. So first, a comment. This is pretty close to me because I spent most of my career helping getting all bioprocess systems approved, including cell and gene therapy. And I helped get the second CAR T therapy approved in 2017. And then 2 years later, my brother was treated with that.
Cool.
And he's, you know, he was, I don't know, 66 at the time. Do the math, but he bought a 50-year-old sailboat. You know, you don't do that if you don't think you're going to live a long time. I happened to attend the chapter in San Francisco of the Frontal Drug Association meeting last spring where there was kind of a panel. In fact, Dr. David Miklos was on it. Do you know him?
Yeah, I do
Yeah.
know. I was on I did a conference with him about a year ago. I was on a panel with him.
He's very entertaining.
Yeah, he is. He's a good guy.
Yeah, but really, I'd say over half, or probably probably 2/3 of the conversation was more about the economics of this. And, you know, let's say that particular treatment, just getting the CAR T cells made is $500,000, and that doesn't include the rest of the expenses. What's your thoughts on that, and how do we mitigate that?
Yeah, oh, that's a great question. It's the big problem in cell and gene therapy right now. And so how that's evolving, again, that was a whole piece of the equation that I couldn't figure out how to fit it in without going way over time. The CAR T version I was talking about, they're taking a patient's own cells, like your brother, editing those and putting them back, and that's called autologous. There's a whole set of companies that are working on taking a donor's T cells, engineering them, freezing them, so you can just pull them off the shelf and put them in a patient. You know, so you eliminate this, take them away for 2 weeks and then bring them back. It just comes right off out of the freezer. And so that's the next evolution. And then the third evolution of that is the CRISPR part. And so now they're trying to actually do that CAR T editing right in the body without even taking the cells out. And the pharmaceutical industry is putting its money on that as being the ultimate answer. But it's too early to tell if that's what's going to happen. Yeah, so across cell and gene therapy, costs are a big problem. It's one of the things that hepatics is close to my heart. A lot of what I've tried to build into the cell therapies we're working on is to reduce the cost as much as possible by making the processes as simple as possible, so you can actually do a cell therapy that doesn't cost $500,000, that it might cost $50,000 or maybe $20,000. Yeah.
Yeah, question more basic level now. You take a cell on the liver, say, and you remove it and edit it, or you leave it in the body and edit it. That's one cell you're putting back in the liver, which has a couple billion cells. How does that affect the entire liver?
So it's a bit of a numbers game. So in the version where you're trying to replace with cells, like we make cells and then we want to put them in back in the cell replacement kind of scenario, it depends on what you're trying to do. If you're treating a single cell defect, a lot of times the thinking is that you only need to replace 5% of the liver, which is a lot of cells, but it's not the whole liver. And in other cases, it would be 30%. So that's sort of the math people think about on that. When you're editing it in the body, I don't know that they've actually figured out with CRISPR what percentage of cells that could be edited are edited. But I bet there are people who are trying to figure that out. That would be part of the development work to make the system more efficient and probably cheaper and easier to use. So that's, in the translation, probably ongoing right now.
Okay.
I think we're going to wrap up for tonight. I think Dr. Scher will be willing to stay for if you have more questions, but the bell for you.